So as Shalimit said, I'm working with AI in lung cancer screening, but why? Why this? Why we need AI to address this problem? Lung cancer is the main cause of cancer-related deaths worldwide in men and women of all ages. And while we know that tobacco smoking is the main risk factor for lung cancer and making primary prevention like tobacco control policies the most effective strategy to reduce lung cancer mortality, we have also another tool for this, that is early detection. Early detection can be achieved via screening, that is a secondary prevention measure by which we target an at-risk population and that is asymptomatic, and we use a test to check which one can develop the disease. So this is to identify the disease at an early stage. And yes. Okay. And the test in the case of lung cancer screening is a low-dose computed tomography, and this was demonstrated by the National Lung Screening Trial for the first time, as well as in the other trials across Europe, like Italung in Italy, LUS in Germany. And these tools demonstrated a 20% lung cancer mortality reduction. But even if this is seems really effective, why we don't have screening trials nationally implemented like it is for breast cancer, colon cancer, or uterus cancer? Because these screening trials were also showing a high overdiagnosis rate, meaning that of the total positive tests that were identified in the program, 96% were not confirmed for lung cancer. So you imagine how many people have undergone useless X-rays or biopsies and other harmful procedures just to know that they had not lung cancer. And also a suboptimal cohort selection, because the main cause of death in such screening programs was not lung cancer, that would be the target disease, but cardiovascular illness, followed by lung cancer and respiratory illness. These three diseases are namely called the big three killers and they are all related to smoking behavior. So all this means that we need to find something to redefine how the criteria, the eligibility criteria to identify the at-risk population, and to better stratify the population to identify who is actually at risk of lung cancer and make the cost effectiveness ratio of a screening program good and favorable to be implemented at large scale. And this is where AI comes in, because I think that we should think about AI not as a substitute, but as a tool, as a magnifying glass that can augment our ability to identify hidden patterns in computed tomography scans that may go unnoticed by the human eye. So it's something, as I said, to augment our skills and not to replace them. And so this is my research project that is about the development of a deep learning framework for personalized post-test risk stratification in lung cancer screening. And I'll briefly explain this long title. So deep learning, because it's deep learning based, as deep learning algorithms have proved very robust in images, medical images interpretation. So personalized because we evaluate the risk of individuals. So we want to maximize the cost effectiveness for the individuals and for the overall population. Post-test because we work on the images. So during a screening, usually lung cancer screening, we have a questionnaire that is collected, that is used to collect demographic data and clinical history data. And also each year, for example, in a national lung screening trial, once per 3 years, a CT scan is performed, and we want to leverage the data that comes from the first CT scan. So we use deep learning to extract data from the images, and we use deep learning also to combine this data with the questionnaire data and leverage them to refine the risk stratification of the individuals, so to better identify who is at risk and predict their evolutionary pathway. And this was not easy because the first problem arose with the data sets, with the management of the data, because we had, fortunately, very big data. This is rare in medical field. You can see NLST enrolled more than 50,000 participants, and we have images for 26,000 of them, and but very small standardization. And this was just to remind how a CT volume is made. So it's a stack of images. Since we had very big data set with very homogeneity, data were collected across 33 centers and 4 manufacturers only for the NLST dataset, we thought to not reinvent the wheel, and we want to reuse an existing standard to standardize these images. And we thought about BIDS, that is a community standard born for neuroimaging, but already extended to other medical imaging modalities like PET, EEG, and also microscopy, for example, so to be outside the neuroimaging field. But it is still not extended to CT imaging. So we started converting a subset of 13,000 volumes. And in the meanwhile, we are collaborating with the BIDS community to officialize the extension. So if some of you is interested, please, I would love to get your insights and feedback to help me finalize this extension. And then once we try to, we settle up for the data management, the problem was to choose a model to extract the biomarkers from the images. And again, to not reinvent the wheel, to not augment further the high entropy of available models we've seen with Gavin before that. The machine learning models in life sciences are a huge amount. We just wanted to reuse existing open source models. And we found very promising tools from very promising research centers and journals like DeepCAC from Harvard, CBIL from MIT. But once we found them, actually, another problem was raised, that was to reproduce the results of such open source tools. And why this? Because data were not fully shared, or the computational environment also was not described, making even slight variations in the hardware attaching the results, the reproducibility of the tool, and also that data processing and statistical analysis were not transparent, and it was not easy to understand how actually the results of the work were obtained. It was like recreating a recipe from a chef restaurant, so but without knowing the ingredients, so the data, without knowing the kitchen configuration, that is the computational environment, and also without knowing actually the recipe, and with many, I would say, chef's secrets hiding behind. And to address these challenges, what we thought once we actually succeeded into the installation and use of this model was creating the containers. So containers are computer science, let's say, abstractions tools that make our software isolated from the operating system and the hardware environment. So like the actual containers, we can move them without caring too much about the context. And we use such container to reproduce our work for the same subset of almost 13,000 CT volumes. So since this talk was entitled Insights from a PhD logbook, I wanted to leave you with what I've learned in this first part of the journey that maybe was already addressed also by Gavin in particular, that medical AI needs validation. We do not want to augment the number of papers, but we want something feasible, something that can make a change in the healthcare field. And to do this, we need to validate this model in different contexts on different data to assess their real-world performance. But validation needs open data and algorithms, because if we have data from different sources, we can develop our models on generalizable data, and also and mostly algorithms, because if we have open and well-documented algorithms, one can use them to perform their independent validation in another context on other, maybe closed data, but they can assess their performance. And also, finally, openness needs fairness, because it's not sufficient to share raw data, raw models, just putting there on GitHub or maybe on other with other identifiers or in other storages, but we need to structure and document them to make them actually reused by other researchers. Thank you.